Hi, let's discuss about medical imaging in biomedical engineering. So this time we want to study about current biomedical imaging technology, and there are various items. So first, I want to show by Vesalius this diagram. He was a physician from Belgium, and in this 16th century, you can see he has made this human brain dissection. This is based on a real dissection. So one can realize inside the brain, there is this ventricle. Without opening it, we couldn't have understood this before. And majority of the biomedical imaging can start with an image. So the image is started from a photography. This is the first photography in 1827 in France. And the B bottom is a modern photography. So these photography or started the biomedical imaging, we can consider in this way. So how biomedical imaging has revolutionized medicine and biology. So our eyes, our retina can only recognize on the surface of the body in most of the time. But however, biomedical imaging allows us to see inside our body and to visualize our biological structure and even functions down to the microscopic scale. So what is image? Image is a representation of measurable properties that vary with spatial position. And oftentimes means if we have multiple images which is taken at different time points, then we can start to understand the dynamic nature of the subject. So image can provide detailed information about biological first as the space, so this is structures, okay? And powerful imaging modalities also provide information, for example, molecular, cellular, and biophysical process, which we mean it as a function. So conventional imaging, we talk about structure, but more powerful imaging, we also want to see the underlying processes that means its function of the biological structures. So, and methods to visualize and quantify these properties from microscopic to macroscopic levels are now becoming available. So image information can be used for clinically for disease, yes, diagnosis, figuring out what kind of disease it is, we can confirm, and for monitoring of a treatment. So even we can use imaging information for monitoring whether the treatment has been taken properly, as well as scientifically for normal and abnormal structure and physiology. So imaging is becoming the most popular and powerful technology we are using for modern medicine. So let me just start with a new imaging technology example. So first, like gene expression imaging. Have you ever thought about this? Based on spatial radioisotopes, which is coupled with genetic methods. So even using radioisotope coupled with genetic methods, we can even visualize gene expression in imaging. So such an example is in advanced PET or positron emission tomography. And the second is a real-time brain imaging during cognitive tasks to guide a neurosurgery. So during neurosurgery, such as, let's say, resection of brain tumor, you don't want to resect a very critical functional part of the brain. So you give the patient to do a relevant cognitive task to visualize the part of the brain. And that technology is called, yes, functional MRI. So, and also right after a heart attack, a patient went right into the clinic and you want to see the action of the heart to identify the injuries. And to map that mechanical action of the heart, with a specialized ultrasound, which we call, because it's hard, so it's called cardiac ultrasound. I will discuss more about this later. So these imaging technologies are essential tools in medicine because they provide a spatial map, enabling our physicians to localize the biological phenomena being examined in space and time. I just have 3 example pictures here. So the left part is a brilliant, very detailed map of inside the body made by X-ray computed tomography, or CT. And second one, this is a black and white, but this is an example of fMRI. Usually based on this, you can find a location that is lighting up for the localized neural activity. And the third is a little poorer spatial resolution. However, it provides a metabolic activity of the brain, which is done by a PET, positron emission tomography. So I'm going to start to discuss about methods to create an image. So to create an image, we need first an energy source, okay? So such as electrons, ultrasound for ultrasonic imaging, light for optical imaging, X-rays for X-ray, CT, or radio frequency for magnetic resonance imaging. And that energy source will interact with the target, for example, like human body parts or organs or cells, and that produces a signal. So I brought one example here. You may be familiar with. You can look at this. This is an example of ultrasonic imaging. And even there's overlaying this red and blue color. This is more functional aspects, such as of blood flow. So this ultrasound, there's energy source is ultrasound. You send it through this transducer, which send the sound wave, and those interface in mechanical structures will bounce off layer by layer, and that is detected by the same transducer, which is converting sound wave into unmeasurable form, such as voltage. So that is a transducer, converting one form of an energy to another form of a signal, okay? So oftentimes, it may be necessary to introduce some kind of material into the subject to highlight a particular structure or function. For example, the barium sulfate for CT. And I brought a picture of here, that the contrast material. So barium sulfate anema makes these large intestine to be visualized with CT so that one can find out if there's any problem in the gut.